Good morning to all. So today we'll continue with the topic of enzymes that we have been going through from past few weeks. We have completed the classification of the we are going to complete about the enzymes. We have completed the classification of enzymes and we have seen all the factors that are going to affect the activity of the enzyme. Then we have also seen how the enzymes decrease the how the enzymes increase the velocity of a how the enzymes increase the velocity of a reaction. So now we'll see how there are, but even though the enzymes are so good, they are so good and they help in so many catalytical reactions, but there should be something that will be inhibiting the activity of the enzymes. All the enzymes are, all the reactions are not good to the human body. So some reactions should not happen. And if the enzymes are there and they are promoting those type of reactions, then they have to be inhibited. Now we'll see what are the various inhibitions of enzyme catalyzed reactions. Inhibitor. Per se, if we define a word inhibitor, inhibitors are any substances that decrease the rate of the enzyme catalyzed reactions. So sometimes they either decrease or sometimes they will completely stop the enzyme catalyzed reaction. So that is what we call it as inhibitors. The ones that decrease or completely stop rate of an enzyme catalyzed reaction. They serve as major control mechanism because though enzymes are very good, for everything a control is very much required. Now these are very important to control the mechanism of action of any enzymes. The mechanism of inhibition is divided into two types. Based on whether it is reversible or irreversible, we divide it into reversible inhibition and irreversible inhibition. The reversible inhibition is further divided into three types: competitive inhibition, uncompetitive inhibition and non-competitive inhibition. So whether the mechanism, whether it is reversible or irreversible, this is further classified. If it is reversible, it is further classified into competitive, uncompetitive and non-competitive inhibitions. The irreversible inhibitions are further divided into affinity labels, group specific and suicidal inactivation. So these are irreversible. Reversible, why do we use the word reversible? It means that when that inhibitor is removed from the medium, the reaction will continue. So it is not causing any damage to the enzyme. That is why we call it as irreversible. And in irreversible, what will happen? The enzyme loses its structure, its function. So once that inhibitor enters into the field, it will completely we won't say it as destroy, but it will completely affect the enzyme in such a way that even though you remove the inhibitor, the reaction cannot proceed further. So that is why we call it as irreversible inhibition. Now we'll see individually what are the types of inhibitions. Reversible inhibitions. Why are they reversible? Because they usually bind with non-covalent bonds. These are not so strong. That is the reason the inhibition is reversible. The activity of the enzyme is restored when the inhibitor is removed. It is characterized by a specific equilibrium between enzyme and an inhibitor. In that way, so enzyme will combine to an inhibitor to form an enzyme inhibitor complex. Like we know that enzyme will combine with the substrate to form enzyme substrate complex. In the same way, an equilibrium is maintained between the enzyme and the inhibitor to form the enzyme inhibitor complex. Now we'll see what you mean by the terms are competitive, uncompetitive and non-competitive type of inhibitions. Now we'll see what is a competitive inhibition. By the term competition, we understand that there is some competition. Who wants to be first and first. So that is a competition. So here we are talking about enzyme is a common one. We are talking about the substrate and we are talking about the inhibitor. So obviously there is a competition between the substrate and the inhibitor towards an enzyme. Now towards an enzyme. Is whole part of the enzyme active, or is a whole the big molecule of enzyme, is it active? No, only a small part of the enzyme that properly involves in the catalytical reaction, that we call it as an active site. So the competition is for the active site of an enzyme. The inhibitors have to bind to an enzyme to cause inhibition. So in competitive inhibition, the competition is for the active site. And the inhibitor and substrate, both of them compete to occupy the active site. And how is it possible? It is possible because the inhibitor is structurally similar to the substrate. And there is no, obviously, when the inhibitor, though it is structurally similar to the product, it exactly fits into, though it is structurally similar to the substrate and it exactly fits into the active site, but that is not a substrate. So that is the reason there is no formation of the product. This goes in this way. The enzyme and the substrate, normally they should form enzyme substrate complex and they form enzyme and the product. So normally the situation has to go in this way. But when inhibitor is added, enzyme will combine with the inhibitor to form enzyme inhibitor complex. Now this enzyme inhibitor complex that is formed, it cannot further, it cannot form a product, it will remain like that. So in this mechanism, what is happening? The enzyme is getting trapped. The enzyme is getting locked as enzyme inhibitor complex. Now if the substrate is forming, you are getting an enzyme back and once again it will go into the reaction. Whereas if the enzyme combines with an inhibitor, the enzyme is locked as EI complex. Now the amount of the enzyme that is present in the reaction will be slowly decreasing. Now, so a low substrate concentration, enzyme substrate, and at low, when the substrate, now we have to think about, now this is a competitive inhibition. So there is a competition between substrate and an inhibitor. Now, if you have more amount of the substrate quantity, if the amount of the substrate is more, so obviously the substrate will win. It will occupy more and more and the inhibitor will occupy less slots. The substrate will occupy more slots, and because the number is very high, there is more formation of enzyme substrate complex. But if the substrate concentrations are very low, then there will be, in the medium you can see enzyme substrate complex and enzyme inhibitor complex and some amount of the free enzyme and very less amount of the product you can see. Now, velocity of the reaction is not changed. Now, how can we come to a conclusion that velocity of the reaction is not changed? Now, just before coming to this, I'll explain this one. High substrate concentrations, all the enzyme combines to form enzyme substrate complex. So, what will happen? Two things is that this is a reversible inhibition. So, the enzyme is not getting damaged. Now, here, substrate and the inhibitor both are structural analogues. That is the reason why inhibitor is sitting into the active site. When the substrate concentration is very high, what will happen? The enzyme, when the substrate concentration is very high, the enzyme will react only with the substrate. It will avoid reacting with the inhibitor because inhibitor concentration is very less. When you increase the concentration of the substrate, the velocity of the reaction will increase and it will go to the Vmax. Now, to understand the kinetics of any enzymatic reactions, you have to be very clear about the Km and the Vmax because Km is a signature of that particular element. So, it gives an idea of the Km will give an idea about that particular enzyme. So, we have to know in all these inhibitions what happens to Km and Vmax. And we also know a graph that exactly depicts the Vmax graph. So, this is called as Lineweaver's graph, where this in this graph 1 by V, that is normally if you remember when we are Michaelis-Menton's calculation, when we are calculating the Km, we had a curve where velocity, only just velocity, it is now 1 by V. Velocity was present on the y-axis and S is present on the y-axis, and X was present on the x-axis, substrate was present, S was present on the x-axis, and the velocity was depicted on the y-axis. Now, when a graph is drawn, it used to come like this, parabola sort of thing. Now, because it was difficult to interpret the inhibitors in that graph, this Lineweaver graph was plotted. it is just nothing but all these things are turned to denominators. So in the y-axis you have 1 by v, and in the x-axis you have 1 by substrate. And this is a v max. Then the where it intercepts with the y-axis, it is 1 minus km, because this is 0, so all these are km values, so it is 1 minus km. Now, the red line represents everything is going on smoothly when inhibitor is not there. Now, once the inhibitor comes, what will happen? Km will decrease. Why the Km is decreasing? Km of the affinity of the enzyme towards the substrate, now we have inhibitor, so that is the reason the affinity of the enzyme towards the substrate is decreasing. As a result of that, Km will increase. In the presence of the inhibitor, the affinity of the enzyme towards the substrate is decreasing. That is why the Km of the reaction will increase. Now, when you increase the amount of the substrate, what is happening? It is reaching to its maximum velocity. So, Vmax is not altered, whereas Km is increased in competitive inhibition. Now, we'll go to examples of the competitive inhibition. Succinate dehydrogenase. Succinate dehydrogenase enzyme become across in the citric acid cycle. So, succinate dehydrogenase will convert succinyl CoA. And this succinate dehydrogenase is inhibited by the enzyme called as malonate. Next comes the sulfonamide and PABA. PABA means para-amino benzoic acid. Now, sulfonamides are antibiotics. Now, how did we come to know that this antibiotic is useful and how both of them are related? The bacteria. For human beings, the folate is obtained from the diet. A little bit of the intestinal bacteria can prepare the folate, but we get it from diet. Whereas bacteria synthesizes its own folic acid. Now, if you know the functions of folic acid, this is very much required for the formation of the nucleic acids. The folic acid is very much required for the formation of nucleic acid. In folate deficiency, the nucleic acid synthesis does not occur. So, the cell development and everything are altered. So, normally what happens in a bacteria, they have a substrate called as methyl hydroxy dihydrobiopterin diphosphate. So, this is a substrate that is present. This combines with the para-amino benzoic acid to form dihydroteroic acid. Now, this dihydroteroic acid will get converted to the folic acid. So, this is how the normal reaction will occur. The folic acid is synthesized in the bacteria, and this is utilized for the bacterial proliferation and growth. Now, the sulfonamides are analogues. These are structural analogues for PABA. Now, what will they do? They will combine with the enzyme that catalyzes with this reaction, and they inhibit the reaction further. So, PABA is not incorporated, and hence there is no formation of the folic acid. So, this compound to get converted to dihydroteroic acid, this compound methyl hydroxy dihydrobiopterin diphosphate will combine with the PABA. Now, sulfonamides being structural analogues to PABA, that will sit on the active site of this enzyme and prevent this reaction to happen. As a result of that, there is no formation of the folic acid. So, resulting in no formation of nucleic acid, no proliferation of the bacteria. In that way, bacteria will perish in the human body. It will not develop, it will get destroyed in the human body. That is how the sulfonamides are used as antibiotics. Next, isoniazid and nicotinamide. Isoniazid is a powerful anti-tubercular that is used in treatment of the tuberculous disease. Now, by what mechanism it was identified that isoniazid to be very good in damaging or in destroying the tuberculous bacteria? That is, it interferes with the formation of nicotinamide. Nicotinamide means NAD. If you know, when we come across like in your intermediate, you might have covered the glycolysis and all that, where NAD and NADH, all these are NADH, FADH2, all these are energy. So, when they enter into the electron transport chain, they produce ATP. So, these are reducing equivalents that will help in formation of the ATP. Now, if their synthesis is affected, obviously the bacteria is not going to survive. because from the NADH plus H plus you'll be getting 3 ATPs, and from FADH3 you'll be getting 2 ATPs. If it is a new nomenclature, it is 2.5 and 1.5 respectively. So what is happening? This isoniazid is preventing the formation of nicotinamide. As a result of that, nicotinamide adenine diphosphate, that is NAD. It is preventing the formation of NAD. As a result of that, reducing equivalents are not formed. The metabolism of the bacteria is altered. That will lead to the death of that particular bacteria. Next, vitamin K and dicumarol are warfarin. So what it was given was vitamin K. The vitamin K, it is a vitamin that will help in activation of the coagulation factors. What coagulation factors are activated by vitamin K? Factors like 2, 7, 9 and 10. So these are the coagulation factors that are activated in the presence of vitamin K, and these get activated and they get involved in the coagulation reactions. So when a drug called as warfarin is given, so this is given as anticoagulant during the surgeries or anything. It is given as anticoagulant. When this drug is given, what will happen? Now to enter into the reaction to help in carbon. Now what are the changes that these factors are undergoing? To these factors, a carboxy group is being added. To add this carboxy group, we need reduced form of vitamin K is required. So, for the inactive coagulation factors like factor 2, 7, 9 and 10, it has to undergo carboxylation to become active. So, this carboxylation is promoted by reduced form of vitamin K. It is promoted by reduced vitamin K. Now, reduced vitamin K in the presence of gamma carboxylase, the coagulation factors are activated and they enter into any of the coagulation pathways. Wherever coagulation is required, that will go into the pathways. Now, what will the warfarin do? And when this process is taking place, reduced vitamin K will get converted to oxidized vitamin K. Now, we have to have another enzyme that will again reconvert it to reduced vitamin K. Now, what does this warfarin do? The warfarin will inhibit the enzyme that helps in reducing the vitamin K. That is epoxide reductase. That enzyme is inactivated by means of the warfarin because it is a structural analog to oxidized vitamin K. As a result of that, reduced vitamin K is not formed, the activation of the coagulation factors will not happen and the coagulation process does not occur. So, this is a function of warfarin that is used as anticoagulant. So, till now we have seen the competitive inhibition and incompetitive inhibition. If the competitive inhibition occurs, what will happen? And how are they therapeutically useful? So most of the antibacterial drugs, anticoagulants, they are synthesized. The basic mechanism behind them is a competitive inhibition. Next, methanol poisoning. This is a very important aspect to remember. Methanol normally, if you have heard, adulterated alcohol. Some people in the villages, they consume adulterated alcohol and this adulterated alcohol is very fatal to the body. What do you mean by adulteration of alcohol? Normal alcohol is ethanol. To that, if methanol is added, because methanol is less expensive when compared to the ethanol, if that is added, that will lead to the toxicity in the body. The main toxicity is mainly because of the formation of formaldehyde. What will happen when any alcohol enters into the body? It is converted to formaldehyde by means of alcohol dehydrogenase. This enzyme will convert it to formaldehyde. Then this formaldehyde will get converted to formic acid or formate. So what will happen? The accumulation of the formaldehyde will create the toxic symptoms to the body. So what is the treatment? How it is? And now you have it has been identified that methanol is poison. Now we have to make something so that this poison can be broken down. So for that, what is done? It is inhibited by giving the ethanol. What will it do? Ethanol will prevent the toxic effects of the formaldehyde. How does it prevent the toxic effects of the formaldehyde? This ethanol will be inhibiting the alcohol dehydrogenase. As a result of that, formaldehyde will be formed. In that way, the toxic effects of the formaldehyde are decreased by means of the methanol poisoning. In that way, so ethanol competitively inhibits alcohol dehydrogenase. As a result of that, methanol will not get converted to formaldehyde. Hence, the toxicity of the methanol is decreased. So, once again to repeat with ethanol, ethanol competitively inhibits the alcohol dehydrogenase. and decreases the formation of the formaldehyde. As a result, of it, the methanol toxicity is decreased in the human body. Next is uric acid synthesis. There is a disease called as gout, where there is accumulation of uric acid in the mucosal tissues, especially in the great toe, in the joints of the great toe. And these uric acid crystals are also get lodged in the kidneys also. So this disease can be prevented. So what is the mechanism of this? Here what will happen? Hypoxanthine will get converted to xanthine, then xanthine to uric acid. So for this reaction we have a common enzyme called as xanthine oxidase. Now this xanthine oxidase is competitively inhibited by the allopurinol. So as a result, what will happen? The reaction will halt at hypoxanthine, and there is no formation of xanthine and there is no formation of uric acid. So competitively inhibited by allopurinol. So by learning the enzyme activities, by learning the enzyme inhibition, one could evolve so many treatments for various disorders. Like we could identify the antibacterial, we could identify the anti-tubercular drugs, we could understand how the poisoning of the methanol can be decreased by giving the ethanol. Then how the gout, that is such a painful disorder, the effects of the gout can be decreased. So all these things we can understand because we know the competitive inhibition. Now, so these are the other enzymes, substrates and the inhibitors and how they are useful. So those are the ones, these are the other ones. Monoamino oxidase. It is an enzyme that helps in catabolism of the catecholamines. Catecholamines like epinephrine, norepinephrine, dopamine, and histamine. All these things, histamine. All these things are serotonin. All these things, especially serotonin. So serotonin, catecholamines, they are degraded by means of the monoamino oxidase. Now, serotonin is a very important ingredient that will help as a mood elevator. So, what will they do in a patients who are suffering from depression? They try to increase the levels of serotonin in the brain. And how do they increase the levels of serotonin in the brain? You cannot give serotonin orally. So, what they do is they will prevent the degradation of the serotonin. Serotonin in the presence of the enzyme monoamino oxidase will get degraded. So, what are they doing? They are inhibiting the monoamino oxidase by giving ephedrine. So this ephedrine will inhibit the monoamino oxidase. As a result of that, serotonin will stay for a longer time in the brain, and that is why it is used in anti-depression. Now, acetylcholine esterase, that is the one that brings about the acetylcholine degradation. So, this choline esterase is important for the degradation of acetylcholine. So, this is inhibited by giving succinyl-CoA. Why should we give an inhibitor for this enzyme? Because it helps in muscle. If acetylcholine is present in the neuromuscular joint for a longer time, it will cause muscle relaxation. So, to prolong the muscle relaxation, to increase the muscle relaxation, or to prolong the muscle relaxation during the surgical process, succinyl-CoA is given so that acetylcholine will stay at the neuromuscular junctions for a longer time. Next, in the cholesterol synthesis, an important enzyme called as HMG-CoA reductase is present. Now, if this HMG-CoA reductase activity is decreased, obviously the cholesterol levels in the bodies will decrease. So, there is a drug called as lovastatin. This lovastatin is structural analog and it will be inhibiting the structural analog to HMG-CoA, and this will be inhibiting the enzyme HMG-CoA reductase. That is why these lovastatins are used to decrease the cholesterol levels in the body. Next, tetrahydrofolate synthesis. As I told you, number one, bacterial cell can synthesize its own folic acid, and the folic acid is very much required for the nucleic acid synthesis. Now, in anti-cancer drugs, what is a cancer? Cancer means uncontrolled proliferation of a cell is called as a cancer. Now, when the cell is undergoing uncontrolled proliferation, that means cell division, it will require more and more amount of the nucleic acids. Now, by giving a drug called as methotrexate that will inhibit the formation of the folic acid, what will happen? There is no folic acid, so there is no formation of nucleic acids. As a result of that, even though the cell multiplies, no proper DNA will be present. Obviously, if the DNA is not proper, the cell has to perish. So, in that way, the methotrexate can be used as an anti-cancer drug because it is inhibiting the tetrahydrofolate synthesis. By inhibiting the enzyme dihydrofolate reductase, the methotrexate can be used as an anti-cancer drug. Next, we'll go to non-competitive inhibition. The inhibitor is structurally different from the substrate, so it doesn't sit into the active site. Inhibitor binds at a site different from the substrate, so it will bind to the normally if the substrate is there, the enzyme should combine with the enzyme substrate complex and form a product. Now, in presence of inhibitor, what will happen? This inhibitor will not bind. This is the site for the substrate. The inhibitor will bind to the any other site that is not an active site, and it will form an enzyme inhibitor complex. One more tricky thing about this is that this because the inhibitor is present at any site other than the active site, the substrate can also bind to the enzyme. As a result of that, you have a complex that is called as enzyme substrate inhibitor complex. So this complex will be formed. Enzyme substrate inhibitor complex will be formed where the substrate has bound to the enzyme at an active site. The inhibitor has bound to the enzyme at any site other than the active site, and there is formation of the complex enzyme substrate inhibitor complex is formed. Now, what is the use of this complex? There is no use of the complex because it is not forming any of the products. Now, the attachment of the substrate doesn't alter the efficiency of an enzyme. Even though the substrate is getting added, the efficiency, the increased activity or decreased activities does not occur. And increase in the concentration of the substance doesn't overcome the inhibition. So, as a result of that, Km is not altered. As a result of this, the Km is not altered in the reaction. So, attachment of the substrate does not affect or alter because already the substrate, the inhibitor is attacking to a site that is other than the active site. So, it is not at all hindering the pathway of the substrate. Substrate will easily fit into the active site. But is there of any use? No, it is not of any use. Even if you go on increasing the concentration of the substrate, it is not able to overcome the inhibition. So, as a result of that, Km is not altered. The affinity of the substrate towards the enzyme and all that are not altered. As a result of that, Km is the same. But Vmax is decreased because even though you are adding the substrate, there is no change in the velocity of the reaction. How it was before when you have added the inhibitor, it is in a similar way. So, the reaction cannot reach to the maximum velocity even if you add more of the substrate. That is the reason the Vmax is decreased. And now, by means of the line-weaver plots, it can be represented in this way. Now, when inhibitors are given, this is without any inhibitor. This is a Vmax without any inhibitor. This is a Vmax with inhibitor. So, Vmax is decreased because it is 1 by Vmax. So, a number higher represents lower velocity. So, in this way, without inhibitor, the reaction would have gone like that. With inhibitor, the velocity of the reaction is decreased, but Km is not changed. As I told you, why Km is not changed? Km is not changed because even if you increase the substrate concentration or anything, the affinity of the enzyme and substrate is not altered. Now, what are the examples of that? Heavy metals or divalent ions inhibit the enzymes with SH group at the active site. Usually, lead will be interacting. Now, if SH is present at the active site, these divalent ions inhibit the enzymes. If the active site has got SH, means sulfhydryl groups are present at the active site. So, usually sulfhydryl group with active site means usually the cysteine should be present in the active site. Lead will inhibit the enzyme ferrochelatase. That is very much required for the heme synthesis, and that will result in the formation of anemia. Fluoride will be inhibiting the enzyme enolase of the glycolysis that comes at the last step, the enzyme enolase of the glycolysis, and as a result of that, it is used, actually it is not used as an anticoagulant, it is used in combination with the anticoagulant. Now, what is the significance of inhibiting the glycolysis and why did we use the word anticoagulant here? Now, the glycolysis is a normal, it is a basic a metabolism that provides energy. That is the basic metabolism, because it requires only one molecule of glucose. From the one molecule of glucose, we'll be getting we'll be getting ATP, energy, and all that. And the importance is that cytosol, the glycolysis reactions will take place only in the cytosol. So as a result of that, even though the cell is non-nucleated also, the glycolysis can occur in the cell. So what is a very important cell that is present in our body? Very important cell that is present in our body, which doesn't have nucleus, and it will depend glycolysis for whole of its energy, for its metabolism, for its function. It depends completely on the glycolysis. It is RBC. So for the in the RBC, glycolysis is only the source of energy. Now what will happen when you draw a blood sample to estimate the blood glucose levels? You have drawn a blood sample to estimate the blood glucose levels in that particular person. As I told you, the RBC, now in that blood sample what we have WBC, will be having RBC. Now WBC will have nucleus, so it will have other methods or other metabolisms can also be there, whereas RBC has only cytoplasm, so it has to depend on glycolysis. Now you have taken a blood sample at maybe 7 o'clock like that, and you want to come to the college and get the sample analyzed in the hospital lab. So you have drawn the blood sample at 7 o'clock, you have packed it properly, carefully, sealed it, labeled it, and you have brought here. Now what will happen is, and now what you have done was, when you have taken the sample at 7 o'clock, immediately you have analyzed the sample at some other place. So the same sample was analyzed after drawing the sample within 5 to 10 minutes, and after almost exactly after 3 hours. Now when you look at the glucose value in the sample that was analyzed within half an hour is higher when compared to the sample that was analyzed 2 hours later. Both the samples were drawn at the same time. Why this difference has occurred? This difference has occurred because the RBC will depend upon the glycolysis. The RBC will be only depending upon the glycolysis. As it is, because it depends only upon the glycolysis, it has utilized most of the glucose. The RBC has utilized most of the glucose, and other cells will also utilize the glucose, like WBC and everything. So what we can do, best thing that we can do is inhibit the glycolysis process in the blood itself. So as a result of that, when you are drawing the blood sample is poured in a tube that contains anticoagulant, along with that antifluorates are also present, that is antiglycolating agents are present. So in that way, the blood cells will not utilize the glucose when you add fluoride in the blood sample. EDTA is one of the anticoagulant. This inhibits the enzymes with calcium and magnesium. So those are the enzymes which require calcium and magnesium are inhibited by EDTA. So that is why it is also used as anticoagulant. Next is uncompetitive inhibition. What do you mean by uncompetitive inhibition? In uncompetitive inhibition, the inhibitor will bind to enzyme substrate complex only. It cannot bind to enzyme separately. It does not have any affinity towards the free enzyme. It will bind to enzyme substrate complex only. So in that way, normally the enzyme and the product will combine. This enzyme and the product will combine, and they will give rise to, the substrate and the enzyme, both of them will combine, and they will give rise to an enzyme substrate complex. That complex will dissociate into product, into enzyme, and into the product. Now this enzyme will once again go back to the reaction to happen. Now when you use an inhibitor, what will happen? The inhibitor will sit at any site other than the active site, and it will inhibit be inhibiting the reaction. So in uncompetitive inhibition, what happens is that the inhibitor does not have any affinity towards the free enzyme. It will only bind to enzyme substrate complex. It does not have any affinity towards the free enzyme. So in this reaction, what will happen? There is decrease in both Km and Vmax. Both of them are decreased because the affinity of an enzyme, the product is not forming. So as a result of that, there is decrease in the Km because the inhibitor is not at all affecting the active site. and the Vmax is also decreased because how much amount of the substrate you add, because the enzyme it is binding to the enzyme substrate complex. How much amount of the substrate you add, how much amount of the free enzyme you add, even then also it does not reach the maximum velocity, because enzyme substrate complex is not getting dissociated to enzyme and the product. It is becoming enzyme substrate and inhibitor complex. So that is the reason in uncompetitive inhibition there is decrease in both Km and Vmax. It is common in two substrate reactions. So these type of inhibitions we commonly see in the reactions where two substrates are present and you have one enzyme. Intestinal ALP by means of the phenylalanine. Intestinal ALP is inhibited by means of the aromatic amino acid phenylalanine. Next, once again to rebrush, we'll see what is a competitive inhibition, what is non-competitive. So in competitive inhibition, there is competition for the active site. So enzyme will combine with the substrate to form enzyme substrate complex. It can bind to inhibitor to form enzyme inhibitor complex. Now as a result of that, what will happen? The Vmax is not going to change, but Km is increased. Next is non-competitive inhibition. In non-competitive inhibition, the inhibitor can either bind to the substrate, or this enzyme inhibitor can either bind to the substrate or it can bind to the enzyme substrate complex also. So as a result of that, what will happen? Even though you increase the concentration of the substrate, the reaction will not reach to Vmax. So as a result of that, Vmax is variable and the Km is the same. Now in another graph, uncompetitive inhibition, you see that the enzyme, it is binding to the enzyme substrate complex, and it is binding to the enzyme substrate complex. We can see that there is no change in the Vmax. Vmax is not formed and there is no change in the Vmax. In uncompetitive inhibition, we can see that it is not how much, because it is binding to a site other than the enzyme, other than the active site. It can never reach to the maximum velocity. So there is decrease in the Vmax, and the affinity of the enzyme towards the complex is also altered. So this is how the reactions will take place. So in this, there is decrease in the Km value and there is increase in the Vmax. There is decrease in the Km value, there is increase in the Vmax. Now, so the decrease, so once again to repeat, with in competitive inhibition, Km value increases, Vmax will change, because there is competition for the site. If you increase the concentration of the substrate, the affinity, if you increase that, the reaction can go to the maximum velocity. Whereas in non-competitive inhibition, because it is binding to a site other than the active site, the affinity of enzyme towards substrate is same, it is not altered, but how much amount of substrate you add, it is not going to decrease the Vmax. Now, the Km in case of uncompetitive inhibition, Km value will decrease and the Vmax value will also decrease. Now, irreversible inhibition. In irreversible inhibition, the inhibitors bind covalently with the enzyme. The inhibitors will be binding covalently with the enzyme. Inhibition cannot be relieved even after you increase the substrate concentrations or do dilutions or any other methods. Once the inhibition has occurred, it cannot be reverted back. And the mechanism is same as that of the non-competitive inhibition. The Vmax is decreased whereas Km is not affected. Group specific. These bind to specifically R group of an active site. They bind specifically to the R group. What do you mean by R group? The extra group of the amino acid. So it will bind to always to the R group of the amino of the active site in an enzyme. What is this R group? The amino acids. If you remember the structure of amino acids, you have C double bond O amino group and the extra group that is present, you call it as a R group. So in group specific, it specifically binds to the R group. One example is TPF, that is diisopropyl phosphofluoride. This will be inhibiting the acetylcholine esterase enzyme. How does it inhibit the acetylcholine esterase enzyme? By reacting with the active OH of the enzyme. By reacting with the active site, that is hydroxy of the serine. Next is iodoacetate. The SH group, it binds to the SH group of the cysteine. For the enzyme glyceraldehyde 3-phosphate dehydrogenase. So this is group specific. They bind to the R group of a particular amino acid present in the active site. DPF is a he will be inhibiting the acetylcholinesterase by binding to the OH group of the serine. Then isoacetate will inhibit the glyceraldehyde 3-phosphate by binding with the SH group of 16. Heavy metals like lead, argon and mercury, why these are all of them, they say it as toxic to the body because they bind to SH group of the cysteine. What is the importance? It is very important. How it is important means because by from these inhibitions you have learned that the active site will be having cysteine, the active site will be having serine, or it will be having the imidazole group of the histidine. So to identify the participating amino acids present in the active site, that is the main importance of the group specific type of enzyme inhibition. Next we'll go into the substrate analogues or affinity labels. These are structurally similar to that of the substrate as we have seen for competitive inhibition. These are highly reactive and covalently block the enzyme. They are not like that, but these are highly reactive and completely block the enzyme site. Next, the examples are tosyl phenyl chloromethyl ketone. These are substrate analog for chymotrypsin and that are very much required in digestion process. Next, 3-bromoacetal phosphate, substrate analog of triose phosphate isomerase and DHAP. So this reaction is present in the glycolysis. So it is a structural analog of triose phosphate isomerase that will be converting 2,3-BPG to that will be converting 1,3-BPG to dihydroxyacetone phosphate. So for that reaction where DHAP will be getting converted to 1,3, for that this enzyme is required, that is inhibited by 3-bromoacetal phosphate. Next, the examples: penicillin. Penicillin inactivates the bacterial enzyme glycopeptidyl transpeptidase that is required for the formation of the cell wall, that is where the bacteria. So penicillin is one of the first antibiotic that was invented. And how was it invented? It will be bringing about the destruction of the bacteria. How does it cause? By giving the insulin, by giving the penicillin, the glycopeptidyl transpeptidase. So the glucose and the protein, that is the glycoprotein metabolism is affected. As a result of that, we know that the glycoproteins are required for the formation of the cell wall. The cell wall, proper cell wall synthesis does not occur, and this will get completely digested by the intestinal, by the gastric juices. Aspirin. You might have heard this term aspirin, dispirin. You get an ad in which water is taken, a dispirin dispirin tablet is dropped into that water, and it will give some effervescence, and people drink that and completely they get rid of the headache. So this aspirin or dispirin, how does it work? It mainly works by inhibiting the synthesis of prostaglandins. These prostaglandins are the ones that will be bringing about, we call it as, these are grouped under the lipids. So these prostaglandins are very much required for in the coagulation process, in bone differentiation, like that. It has got various activities. So it will be inhibiting for the synthesis of prostaglandins. You require the last step, the initial step, you require an enzyme called as cyclooxygenase. This cyclooxygenase is inhibited by aspirin. In that way, the prostaglandin synthesis is decreased. As a result of that, coagulation factors and are affected. Next, disulfiram. This disulfiram is an important thing that is used in de-addicting the alcoholism, de-addicting the alcohols. How can the alcohols be de-addicted? By giving disulfiram. It is because when alcohol is consumed in the body, when alcohol is consumed by the body, what will happen? The alcohol, in the presence of alcohol dehydrogenase, will get converted to acetaldehyde. From acetaldehyde, it will get converted to acetic acid, and that is how it is eliminated. So what will happen in alcoholic patients, and the fatal effect, the dangerous effects that will result is because of the accumulation of the aldehyde. Now, in the first step, where alcohol is getting converted to aldehyde, that is ethanol is getting converted to acetaldehyde. Further, acetaldehyde getting converted to acetic acid is inhibited. It is inhibited. So that is all aldehyde dehydrogenase. The enzyme is aldehyde dehydrogenase. That enzyme is inhibited. So as a result of that, in the body, the aldehyde cools up. The acetaldehyde cools up, and this acetaldehyde, when it gets cooled up, it will give such a adverse reaction or such a adverse feeling in the body that after looking into that, the people will stop. Now if he consumes once again alcohol, he'll have that adverse feeling. So what will he do? He will restrain from consuming the alcohol. So in that way, for de-addiction of alcohol, disulfiram is used. What will disulfiram do? It will promote the accumulation of acetaldehyde in the body. Because acetaldehyde is getting accumulated in the body, the person will have different type of situation where he cannot tolerate that situation. So what will happen? Because of that, he will not consume alcohol again. Next, N-dimethyl propyl-glyamine. So this is one that will be inhibiting the monoamino oxidase enzyme. As I told you, monoamino oxidase enzyme is required in degradation of the serotonin. To maintain the serotonin for a longer time in the brain, we give the drugs that will inhibit the enzymes that are involved in the degradation of serotonin, so that serotonin is maintained at a higher levels in the body. What is the significance of what is the significance of all these studies? The significance of understanding, why we have to understand, is the significance of the substrate specificity. You are understanding the substrate specificity of an enzyme. So this enzyme, how is it activating? How specifically is it attacking? The nature of the functional group. By looking at that, you understood that the nature of the serine containing the serine, in some of them serine is present, imidazole is present as an active site, cysteine is present as an active site, and we also understood the mechanism of catalytical activity. Next, elucidating metabolic pathways. So we can understand how the metabolic pathways are taking place, like some of the enzymes will be inhibiting the major metabolic pathways, like fluoride will be inhibiting the enolase, all these things. Next, therapeutical applications. Penicillin was identified, aspirin was identified, and disulfiram is identified. Penicillin is an antibiotic, aspirin will be preventing the coagulation, it is an anticoagulant, disulfiram will help in de-addicting the alcoholics. Now, N-dimethyl propyl-glyamine. Now, next comes across an inhibition called as suicide inhibition. What do you mean by suicide? Suicide means self-destruction, destroying one own self. Now, why is that it is destroying one own self? You have given an inhibitor or any substances. That substance, normally it is very unreactive. But what will happen once it enters into the body, into the biological system, it becomes much more active and it will destroy the reaction. So that is why you call it as suicidal inhibition. On binding with the active site, it will carry out catalytical changes of normal reaction, forming high reactive compound which inhibits the enzymes. Examples are allopurinol. This we have come across when we are talking about the competitive inhibition. Allopurinol will be inhibiting an enzyme called as xanthine oxidase by means of competitive inhibition. At the same time, what will happen? The allopurinol, in the presence of the enzyme xanthine oxidase, will get converted to a powerful compound called as alloxanthine. This alloxanthine is very toxic when compared to the allopurinol, and it will completely prevent the formation of the uric acid. Next, 5-fluorouracil. The thymidylate synthase, nucleic acid synthesis. So this thymidylate synthase in the presence of 5-fluorouracil, this also we have covered, this is used in anti-cancer drugs. So 5-fluorouracil in the presence of thymidylate synthase, it will get converted to fluorodeoxyuridylate. It is converted to fluorodeoxyuridylate. So in that way, these are the examples of the group specific type of the inhibition. Next, specificity of the enzymes. Ability of an enzyme to discriminate between two competing substances. Now, as I told you, enzymes are present in the cytosol. You have enzymes in cytosol, you have enzymes in mitochondria, you have enzymes in like that. But in cytosol, you have n number of enzymes. Now, how is one enzyme recognizing the other, its own substrate? Why one enzyme is not combining with some other substrate, like hexokinase and the glucokinase? Why are they binding either to the hexoses or to the glucoses? Why these are not binding to acetyl-CoA or some of the fatty acid chain? So how are the enzymes acting specifically? So the ability of an enzyme to discriminate between two competing substrates is called as specificity of enzyme action. It is very much required because it makes it possible for many reactions to coexist in a cell without interfering with each other. Now, the fatty acid synthesis takes place. The fatty acid synthesis takes place in the cytosol. Glycolysis also takes place in the cytosol. So, in fatty acid synthesis, you have many enzymes. Glycolysis also many enzymes. But both the reactions are occurring parallelly and they are not interfering with each other. So, that is done mainly by the specificity of the enzyme action. So, how do we define different types of specificities? There are 4 types of specificity: absolute specificity, group specificity, reaction specificity, and stereo specificity. What do you mean by absolute one? Absolute one means it will act only on that particular substrate. Like glucokinase will act only on the glucose. It will not act on fructose. So these fructose or any other component. So that is the absolute specificity. Now when we use the word hexokinase, hexokinase is one that can act on any of the hexoses, whereas glucokinase will act specifically on the glucose. Another example is lactate that can act only on the lactose. So that is absolute specificity. Next is group specificity. Acts on specific bond with specific groups like phosphatases. In that way it will identify that. Now the phosphatase acts in, now the enzyme phosphatase will remove the phosphate group from any substance. So it will identify the phosphate group and it will remove the phosphate. Trypsin, the enzyme that will break the link between lysine, or if the lysine is attached with any other amino acid, if arginine is attached with any other amino acid, that is broken down by means of the trypsin. So trypsin identifies the peptide linkage here, and here the phosphatase is identifying the phosphate formation of the phosphates. It is identifying the phosphate here. The trypsin is identifying the peptide link that is present between the 2 amino acids. It is specific to 2 amino acids, that is arginine and lysine. Next, chymotrypsin. The chymotrypsin identifies the aromatic amino acids and there it catalyzes the reaction. It will bring about the proteolysis. Next is reaction specificity. In the reaction specificity, what will happen? The enzymes that are reaction will be taking place. So like catalysis, only one, they catalyze only one specific reactions, like pyruvate. In the presence of pyruvate dehydrogenate, it will form acetyl-CoA. Now the acetyl-CoA that is formed, once again it cannot get converted to the pyruvate in the presence of the enzyme. So reaction specificity. The reaction will happen only on the one side. The pyruvate in the presence of pyruvate dehydrogenase complex will get converted to acetyl-CoA. Lactate in the presence of lactate dehydrogenase will get converted, pyruvate will get converted to the lactate. So this is reaction specificity. Here the type of the reaction is dehydrogenation. Here also dehydrogenation. Here the reaction is carboxylation. Pyruvate will undergo carboxylation to form oxaloacetate. Then transamination. There is transfer of amino group. Pyruvate is getting converted to alanine. So these are reaction specificity. It is specific to one particular reaction. Next is stereo specificity. It will act only on one isomer. Like L-amino acid oxidase will act only on the L-amino acid. D-glucose oxidase will act only on the D-glucose. Salivary amylase acts on alpha-1,4 glycosidic linkage. It will be acting only on alpha-1,4 glycosidic linkages. So, till now we have completed the inhibitions. Inhibitions are very important. They can come as an essay for you. We have seen inhibitions. Inhibitions are of 2 types, reversible and irreversible. Reversible because the inhibitor and the enzyme bind with a non-covalent in a non-covalent bond, so that is why it is reversible. Irreversible, it binds with a covalent or very strong bonds, that is why the reaction is irreversible. And in the inhibition, reversible inhibition, we have seen competitive, uncompetitive and non-competitive. And in the reversible inhibition, we have seen affinity labels, stereospecificity, all these things we have seen in the irreversible inhibition. Now we have seen the clinical significance, how these inhibitors are used. This knowledge about the inhibition is used clinically to create antibiotics, to generate or to synthesize antibiotics, to synthesize anti-cancer drugs, and also to use as anticoagulants to improve. So to improve the life and all that, the property of the inhibition is understood and it is therapeutically applied to form antibiotics, anti-cancer drugs, anticoagulants, all these things. And all these things are formed by understanding the property of the inhibition. Next we have seen specificity, how the enzymes are specific. So they will be having group specificity, they can have stereo specificity, they can have substrate specificity, like that. They have some specificity because of which they do function. So with this I am closing today's class. Tomorrow we'll start with the regulation. Remember, the inhibition is very important. It can come as an essay.